Thank you, Simon. I appreciate Simon and Mark for inviting us to this event. It has been many years that we have been participating in these events, and we certainly appreciate the opportunity today to talk about our approaches to how develop amorphous solid dispersions and modified release with minimal amount of drug when we are right after discovery. And we want to see how we can make this the smoothest possible for transition from one stage to next. So we are all familiar with the early stages of development when the drug is being discovered. Obviously, there are different rules of thumbs in here, but what we know is that there are tens of thousands of molecules that have to be discovered. From those that have been discovered, we have to do hit selection, candidate optimization, and select a drug for a preclinical or phase 1 or phase 2 development. So we are talking about tens of thousands of drugs that have to be discovered through those discovery pathways, and then from those that have been discovered, how we can make sure that we can do these formulations that these early stages of development, without using so much of drug. Because when we are at the 10,000 level drug and we come to the 5, that's when only milligram amounts of drug is available. So the issue that we have always with the formulation is how we can do these developments with only 100 milligram of drug. There are many ways to do this. There are many technologies to do this kind of work. And of course, there are many providers that provide equipment to do these kind of early stage screening for amorphous solid dispersion. The question is always how we can do this most efficiently and not to have to revert back later on when we take those 5 compounds into development. We are talking about anywhere from 6 to 10 years time taking from a discovery of a new drug, a new chemical entity, all the way to its commercial manufacturing. So basically, it's like American Idol for singers and drugs. So there are many drugs out there that can be discovered. Which one is going to win the game and is going to go and become that commercial product that we are looking for? As I mentioned earlier, during this process of discovery, we are talking about identifying the right target, validating the target, doing our lead optimization, hit to lead optimization. All these processes have to happen. Then we have the pre-formulation, preclinical studies, animal studies, IND, enabling studies, which is our focus right now in this talk. And then IND application, phase 1, phase 2, phase 3, commercial and post-approval. Many years of development and process has to happen until a drug reaches to that place. So how we can build quality into a product development and ensure that the final product is going to be stable, commercially viable, and helpful for the patients who are waiting for it. So when there isn't a lot of drug available, there are many ways to do this. One is, before that we go in the lab and start working, is the paperwork. And then the next step is miniaturization, when there isn't a lot of drug available. So how we can do this efficiently comes from the fact that there have been lots of studies done on the paper exercise, and we know that over 70% of drugs in the development right now are poorly soluble and or poorly permeable. For all of those drugs, actually, amorphous solid dispersion can be screened and can be utilized as a way to improve solubility and bioavailability or even permeability. So the BCS classification has been utilized for many years as the basis for minimizing the number of experiments, understanding the drug characteristics as far as its bioavailability and solubility and permeability goes. So basically doing a first paper exercise and understanding if the log P is in the right place, maybe a lipid formulation is going to be good if the proper interaction can happen between the drug and the polymer, maybe an amorphous solid dispersion can be utilized. But that being said, further, once we have that selection, once we understand if the drug can be enhanced for bioavailability through amorphous solid dispersion or any other technology, we have to go into experimentation. So further to that process, most recently, as of 2010, there is the developability classification system in place of biopharmaceutical classification system on the fact that not only dissolution is a problem, but also not only the dissolution is may be a problem. I have put this on silent, but still it is ringing. Sorry about that. Maybe it's my watch. I didn't think about that. Okay, do not disturb. Yes, so when it comes to developability classification, what the team, Butler and et al., came up with a new way of looking at the bioavailability enhancement and solubility and bioavailability issues. They looked at hundreds and hundreds of drugs that are already in the market or in the development, and they saw that even in the BCS2, where the solubility is low but permeability is high, you can have drugs that may have solubility limit issues or dissolution rate issues as far as bioavailability goes. So for those systems, they came back and they further analyzed the drugs and showed that if the solubility is an issue, if the saturation solubility is an issue, then only a fraction of drug is going to be dissolved, and then nothing is going to be absorbed. It is going to pass through the GI tract. And then if the solution rate is the problem, that means that you need to increase the surface area and try to make sure that the drug is going to be absorbed faster through the GI tract, and they are not limited by solubility by itself, but they are limited by the rate of solubilization. So based on that, this paper exercise is telling to us that if we can identify the permeability and solubility of the drug, and further to that, identify if the drug has a dissolution rate issue or the solubility, dissolution rate issue, dissolution saturation solubility issue or dissolution rate issue, then we can utilize different technologies. So amorphous solid dispersion is going to work only if you have a saturation solubility issue. Lipid-based formulations and so on can be utilized here. Micronization and nanosuspensions are going to work only if you have a dissolution rate issue and not saturation solubility issue with regards to your drug product. And they further showed that using model drugs that are already developed and commercialized. Further to that, when it comes to developing new chemical entities, modified release can be a way to improve bioavailability and overall problems with the drug product. Why is that? Because of the fact that if we are developing drugs that are going to be unstable in the acidic conditions of stomach, we need to make sure that we have protection for those. And that can be an enteric system, or that can be even a sustained release system, delivering the drug to the right area of the GI tract. But what is happening is that when we are at these early stages of development, we don't have hundreds of grams of drug available. So our approach is to develop a peptide for oral delivery, or a protein or enzyme or whatever with minimum amount of drug available. And that is when the miniaturization is going to be needed. And that is what we are trying to present today to you about how we can develop these products for pharmaceutical industry right after discovery, when micrograms level of formulation is needed, all the way to hundreds of kgs to metrics of tons. How I can do this screening efficiently at these early stages of development. So when it comes to amorphous solid dispersion, before that we get started, obviously it's all about miscibility. We are looking for miscible pairs of drug and the polymer. How we can do that is a simple fact that like dissolves like. If we have the solubility parameters for the drug, the solubility parameters for the polymer, then we can do a rational selection of which polymer and drug to mix and match. Then we minimize the amount of drug that is needed to run this screening study. So again, first is going to be the classification that we talked about. Look at this. Look at the solubility. Look at the permeability. Decide if it is a DCS2 or DCS2A or 2B. Then decide if amorphous solid dispersion is the right way to go. And further to that, before still using any milligram or microgram of drug, basically zero utilized yet, do this in silico model, see which drug and the polymer combinations are going to be miscible, then run your miniaturized prediction. And of course, there are many ways to do these miniaturized predictive experiments, but film casting certainly simulates what is happening with spray drying. For hot melt extrusion, there are many ways to do that. Of course, Daniel is here as well, and he has his equipment as well that he utilizes to do these hot melt extrusion experiments at various early stages of development when melt prep can be utilized for that, as a matter of fact, where you don't have a lot of drug and you can utilize minimum amount of drug and polymer combination to make those amorphous solid dispersion for hot melt extrusion. But that being said, that miniaturization is done with many places where we can do that. This can be done in-house by anybody. But what advantage that we have at BioDuro specifically, and my team, is that we have our own animal study site. So our in silico model plus that first screening in the lab for amorphous solid dispersion plus animal study, that's 4 weeks. So we screen all the polymers, we screen all the technologies that are out there, including lipids or and nanosuspension or micronization. Any technology that is out there for dissolution rate limit systems or solubility rate limit systems, we can do that within 4 weeks of development, including one round of animal study. And then if it worked, great. If it didn't work, we do another 3 weeks of development, another round of animal study. So we are talking about 8 weeks where we can screen all these formulations right at the discovery stage with milligram amounts of drug utilized, because we do that paper exercise first. Before that, we mix and match products. So when we talk about amorphous solid dispersion, of course, we are all familiar. I'm not going to preach to the choir about the fact that what we do is we break the crystal lattice of the drug, we disperse it within the polymer. But one myth that I have always heard about amorphous solid dispersion is that we have to just worry about the TG and have the polymer with highest TG. Yet what is important to know is that if TG is the only tool that you are utilizing to stabilize your system, a moisture absorption, any kind of change in TG is going to reduce that TG, and you are going to have recrystallization. So in addition to the TG, what we recommend is to look at the structure of the drug, to look at the structure of the polymer. and decide if there is going to be any hydrogen bond, ionic interactions, or any other kind of interaction happening between the drug and the polymer that can further stabilize the system. So basically, we are physically attaching the molecules to the polymer and then stabilize the system through ionic or hydrogen bonds or polar or dipole interactions. So this is what we are looking for when it comes to our initial selection of the polymer and drug combination. So we don't just mix and match randomly. We want to make sure that that interactions is there. So two technologies that are utilized for currently amorphous solid dispersion. One is spray drying, and the other one is hot melt extrusion. Both can utilize the same actually kind of modeling to find the miscible pairs of drug and the polymer. But when it comes to experimental work, what we need to look into is that, here I'm giving the analogy about the water and how it dissolves sodium chloride and how it dissolves sugar or sucrose. So as we know, crystal structure of salt, sodium chloride, is very strong. In order to break that crystal lattice, we need 801 degrees centigrade. But I can take the same crystal lattice of sodium chloride, put in water, and dissolve it at room temperature. I'm breaking the crystal lattice that requires 801 degree to break using heat at room temperature with simple ionic interactions. And the same with sugar. When we put the sugar in water, we are relying not on ionic interactions, but on hydrogen bond to break that crystal lattice. So why not to look at the polymer and the drug in the same manner and look at a drug that is going to be amorphized within a polymer matrix using hydrogen bond or using ionic interactions. So I worked at Evonik. At Evonik, we had a polymer called, and still they have it, Oidogate E. Oidogate E is ionic. It's positive in ions. And if you take any acidic drug and you put inside Oidogate E, you will see that it's going to miscibilize very quickly, actually. Many of those drugs, they can miscibilize easily within that polymer. You get ibuprofen and you put with Oidogate E at room temperature, come back in a month, it's going to be one phase. Because actually solids can dissolve into each other, it's just a matter of time, when that dissolution is going to happen. So we need to not only look at what polymers have been utilized for amorphization of other drugs. Let's say Calitra example comes all the time. Yes, and in that case, certain polymer is being used, VA64, but that is because that polymer and those drugs, they are able to create that interaction to create a miscible pair. It's not just the TG that helps them to stay there. It is also hydrogen bond interaction between PVP and those drugs. So how I can find the sweet spot for solubility of a drug and the polymer? So that equation was already in the literature. Flory Hoggins, Flory got the Nobel Prize for that equation that says a polymer is going to dissolve in a solvent if the Gibbs free energy minimized for that concentration of the polymer in the solvent. So what we are doing in here, we are reversing the roles between the drug, which is the solute in here, and the polymer, which is the solvent. So if we solve this equation based on the chi that can be calculated, we can find where the Gibbs free energy is minimized, and that's the maximum concentration of drug that is going to saturate your polymer. So you want to start your experimental plan, you don't start in the dark, you start where the predictions are. Is this validated per pharma? No. But this is the place where you start if you have only 200 milligram of drug, for including 2 rounds of animal studies. So if you have only 200 milligram of drug, you don't have the luxury of screening all the polymers. And many of the people that we worked with at BioDuro, they are on the early stages of development, small companies looking for investment. They have screened tens of thousands of compounds. They have identified one compound that is showing everything perfect. The only problem with it is that it's dust, brick, sand, whatever we want to call. Actually, I had a client, I said, your drug looks like, I use the terminology that everybody is using. I said, looks like dust and brick, this one. It's not going to dissolve. He's like, what are you talking about? This is a very expensive drug. And I said, okay, so it's diamond, so it is even less soluble. So if you have a very poorly soluble drug, and you have and that drug is actually working on the target, but is not able to give bioavailability that is going to wow the investors, that's when you want to make sure that you do this screening when you have only a few milligrams of the drug, and you show to investors that, yes, this works. Let's go to next step, and then let's go to scale up and make metric tons of this material rather than just a few hundred milligrams. So once we identified which drug and polymer combinations to use, and we did the animal study, as you saw earlier, with regards to showing that the bioavailability is there, then we have to go to scale up. 15 years ago, when I was in this field, everything was hot melt extrusion. We were just, most people, it was the easiest way to go. We were utilizing hot melt extrusion more than spray drying. Nowadays, everything is going through spray drying first, and then at the right time, we switch to hot melt extrusion if it works, because it is less expensive technology, a lot easier and straightforward than five-story building to fit a spray dryer. But that being said, that screening has to happen at these early stages of development, and we have to make sure that the right process is developed. So once the formulation is identified, everything else is about process development, about creating a reliable process that is going to give us the best formulation and best product that can be utilized with patients. When it comes to controlled release applications, when you are working with a peptide, a protein, and you have only just few milligrams of the drug at these early stages of development, one way to do it is, of course, putting the drug in a capsule and then coating the capsule. Unfortunately, many of these capsules, they are not going to be fully filled. And if you coat them, then there is air bubble inside. It's going to float. It's not going to pass properly through the GI tract. And it's going to cause you many false negatives from those studies that you're going to do with an enterically coated capsule. So in those cases, especially when you want to try, actually, we have done this. We have utilized this in rats, and it works. If we make mini tablets rather than capsules, you have just a milligram amount of a peptide. The only way to deliver it right now is probably injection, and who wants injection? Oftentimes, I worked in alchemies too. If you back in early 2000, the technology have improved with injectable controlled release systems nowadays. The needles are a lot finer, but still, when you see that garden hose through which you have to inject some microspheres in you, and you see the difference with the tablet or capsule that you have to take, I go with the capsule right away. But nothing against injection. If there are some drugs that are not going to work with oral application, but we have in pipeline for many other companies that we are working with, that they have peptides that they want to do oral delivery through a much more patient compliant way. If you are looking to develop a peptide and you are at these early stages of development for oral applications, or you want to do just a side study and see if, how is the bioavailability for oral versus injection, then this is the mini tablet we have seen is the best option for these early stages, because basically you can manually, or in a bag or whatever, Make your blend, compress mini tablets. These mini tablets are 1.5 millimeter in diameter. They pass through the even rat GI tract with no problem. If not, you can just put them, multiples of them, into a capsule, immediate release capsule, of course, and then dose into dogs or monkeys or mini pigs. Of course, mini pigs are going to be a lot more relevant for many of these biologicals and peptides and protein delivery. But if you do that, then you can utilize only milligram amounts. We are talking about maybe altogether 300, 400, 500 milligram of drug that you need to make mini tablets. And then you can coat them in a conventional coating machine. You don't have to even scale it down. Why is that? Because we can make simply the active a different color than placebo, coat the whole batch inside an actual coating machine, and then handpick the active from the collection, and then dose those into animals, and don't worry about the capsule direction and is it going to dissolve the capsule or not, and so on. So this is how we miniaturize early stage. So my point at the beginning of this talk was how I can close the gap between discovery and clinical predictions and clinical work with minimum amount of drug. So paper exercise is essential. Do that paper exercise first, select the right combinations, make sure that you have the, for amorphous solid dispersion, you have the right pair of drug and the polymer and so on. For these kind of applications, of course, we are going to add other ingredients. If I have to add a muco-adhesion component, it can be added inside the mini tablet, or it can be coated. If I have to add any permeability enhancer, they can be part of the tablet process as well. And then once you mix all of that, you can do the coating. So let's talk about case studies. This is a case of, again, a place where we had to do amorphous solid dispersion compared to other technologies. So we were asked, nanosuspension, micronization, I don't know, everything that is out there, if one after the other. So when there was milligram amount of material available, we did the amorphous solid dispersion. But later on, when there was more drug, people asked the question, I didn't screen nanosuspension. Maybe there is something out of there for me to win or whatever. So we did all these technologies with this project. And we were able to look into those technologies and see which one is going to work the best. So here is the solution that we got from the system that is amorphous solid dispersion. We saw that the Calydone VA64 and TPGS gave the best solubility. This was before that we did this. We did, of course, our calculations. The in silico model, we found the right polymer, minimized the amount of drug, and we came up with this combination that worked the best. If you look at the Calitra formulation, which is Norvir and ritonavir, both of them, they are BCS4. They have to be dosed together. So we know that those two drugs together, they get absorbed much better than each one of them individually. But on top of that, if you look at the even Calitra formulation in the inactive ingredients list, you will see that the surfactant from soft gel is still there. So this product was previously a soft gel. It has changed into an amorphous solid dispersion, but they still kept the surfactant, and that's what we saw in here. That adding VA64 to this compound actually helped increasing bioavailability of solubility of this drug, and later on we showed that it increases also bioavailability. When you do, of course, early stage development of amorphous solid dispersions and you have enough drug, then you, in addition to the conventional dissolution, you develop also non-sink dissolution systems because of the saturation that is going to happen with these systems. And again, further to that, when we had enough drug and we were able to do non-sink dissolution, we further showed that actually that combination works best. Then we tried all of that in animals for rodents. And oftentimes we use mice rather than rat because when you want to run rat studies, you need a lot more. let's say maybe 400 milligram of drug you need versus 200 milligram for a mice study. So once we did that, and this is the bioavailability that we got from the system, all these systems, as you can see, the solution in PEG water, nanosuspension, and STD and HME, and the tube results. The tube refers to that the microtubes that we saw earlier, where we do film casting and evaporation of the solvent. As you can see, the evaporation of the solvent using film casting process gives the same area under curve. Basically, it's a validated system showing you that you can get the same bioavailability from that system as spray drying. So this means that the system is validated for us, and this is what we utilize at these early stages of development to develop our formulations. This is a poster that we published further at AAPS. We have another one at CRS, and of course, Daniel Treffer, and we will be at CRS as well. Thanks for inviting us for that workshop that is going to happen before the CRS as well. We will be there, and we will talk and present another poster about solution engine, where we have been able to develop, again, a solubility enhanced system using minimal amount of drug with actual PK profile from animals. So for modified release, again, those mini tablets are helpful. We have done this again and again. We are able to miniaturize controlled release system to very small scale. Again, milligram amounts of drug utilized, and then by actually playing with the weight gain and the solvent or whatever that we are utilizing, we are able to get the system to respond to the solution rate control in those processes. Further to that, we again applied similar systems at the early stages for this product, where we developed mini tablets when there wasn't a lot of drug available. Following that, we were able to switch that to a multi-particulate. Of course, there are sellets and so on and so forth that you can clearly control the particle size of a controlled release system and surface area, and then you can do how many layers of coating you want around that. So here we actually developed the product that has multi-layer 2 drug in one combination that is allowing us to release the product at the right conditions. So we developed that process, we validated, and actually this product is commercial at our site. We utilized, again, the QBD concept. So as you can see, you start from milligram, microgram levels of experiments all the way to metric tons. The same principles are going to apply. You have to do your risk assessment. You have to understand what are the risks if I use a capsule versus a tablet, and so on, until you find the right formulation, the right combination, encapsulation, and then further to that, during your process, what you want to show is that you have good control on the weights, good control on the coating, and good control on your overall product performance. So we have, in order to get this done, we had to validate our process. This is to show you that what we are talking about from the microgram level all the way to commercial is a true correlation, and this is happening already within the pharma industry and the services that we offer. So overall, just for your information, we have at BioDuro, we are a collection of 4 different companies that grow on their own. We are continuing those legacies within our company. So one of the legacies that we started was back in 1996 at UT Austin. The original company was founded. So we started our company with hot melt extrusion. Later on, we added spray drying and other capabilities there. So the company is located in Irvine, California. By all means, you have my email and my information at the end available for this presentation. And I look forward to speaking with you and hearing any questions that you may have. Thank you.
Okay, thanks very much, Firouz. We've got a couple of minutes before the next session. Any questions you'd like to follow up with? Yes, Carol.
Hi, Karu. Nice to see you again. Yes.
So great presentation, thank you.
Thank you.
Just quick question, what's the timeline? I think I noticed 8 to 10 weeks from start to finish. This is to get up to phase 1, or what is this,
This is
8 to 10
to
weeks?
this is to do screening. So basically, what we are doing with minimal amount of drug, we give enough information to our clients to go back to their investors and say that this diamond that we have and it is not dissolving has a chance. So it is not, it doesn't include stability. There is no way that we can do stability studies and so on at these early stages with the amount of drug that is available. But we do also ASAP studies right after that. So minimum amount of drug, minimum amount of time. We can do ASAP studies for stability and quickly show within a month that, after that, the stability is there as well. So it doesn't include stability, but 8 to 10 weeks is screening, amorphous solid dispersion, SEDS and SMEDS, micronization, and nanosuspension. We do all of that in that.
And the animal studies.
And the animal studies, yes.
Okay, thank you.
That's because it's an internal transfer. It's an internal work. So, kind of an adaptive, clinical trial that some other companies, they do with humans, we do with the animals. Yes.
Yes, Corbinian Lübmann speaking from Serum Pharma. Thanks for your presentation.
Thank
I
you.
was wondering, now you presented, of course, only about amorphous solid dispersions, but your company offers all of these enabling technologies. Could you put a rough number on how many of those are actually falling into DCS class 2A and how many are actually then going towards an amorphous approach?
Yeah, so I mean, if you looked at that table that I had for even that client, we showed that the drug was actually better bioavailable using nano suspension. So meaning that the client agreed that that drug has solubility rate limit clearly from nano suspension. But when they looked at, basically no bioavailability from oral system, but, significantly better bioavailability from amorphous solid dispersion and potential for a tablet versus a liquid nanosuspension, their decision came back to us and said, okay, it is double bioavailability with nanosuspension, but I don't want that. You have enough bioavailability for me to go with a tablet. So we went for it with a tablet for that one. That one is a phase 2 product right now that we are talking about. So oftentimes, actually, you're right, that we show the validity of the nanosuspension and everything else to our clients, but the easier route, the more economical route and convenient way to go has been for us amorphous solid dispersion. So 70–80% of cases that we do, despite the fact that we screen everything else, ends up to be amorphous solid dispersion as a way to go for the phase 1, phase 2.
Very, very interesting. And I have another one. You mentioned during your presentation that initially people always want to go towards the spray drying, but then later on they want to go into a more cheaper process. Could you comment on why there is such an interest in the spray drying itself?
Because again, the amounts of drug that you will utilize is a lot less, and there is no heat involved, significant heat. The exposure to heat is a fraction of a second. With extrusion, always, people have hesitation. Oh, it's going to be too hot. It's going to burn it, whatever. I don't want to, deal with any impurities and adding antioxidant or whatever. So they are not going that route because they want to get to the phase 1 with clean system, with no questions about, was it any problem during that process or not. And then once they have clinical data, now they are relaxed. They can come back and revisit and show that actually hot melt extrusion is, worth it as well, and then eliminate solvents. 20 years ago, if you told me that this spraying that I'm doing is 98% solvent, we have products that they are 98% solvent. Basically, there is every kg, there is only just few gram of it is the drug when you spray. But yet, it is worth it nowadays because they show that there is no other way for that drug to survive, to go into the GI tract or survive the heat from hot melt extrusion. So spray drying is still going much faster than hot melt extrusion, despite the fact that the systems are very, very dilute. when it comes to actual solution that you are spraying.
Great, thanks.
Yes.
Thank you, Firouz, for the nice presentation. I have a question on the last statement that you made. You know, the preference to spray drying as compared to HME because of heat, is that a perception issue or is that a real issue according to your opinion?
I think at the phase 1 stage is a perception issue. Phase 1 stage, most of our clients, they don't want to deal with any risks or whatever that can be associated with high heat and degradation and byproducts or color change or whatever else. So phase 1, we go with perception, which is spray drying is going to be less questionable, more clean product at the end. But when it goes to phase 2 and phase 3, we come back and actually revisit. Every client that we have, we always recommend to go back and revisit hot melt extrusion because the system can fit. This room is too big for that. The biggest hot melt extruders out in the market. This whole building is too small for spray dryers. Yes. So and there are a handful of people that can do spray drying versus hot melt extrusion can happen everywhere. So they do their risk assessment. But oftentimes, in the past, what I'm trying to say is that when it was 2% solids content in a spray-dried system, everybody was saying, oh, that doesn't make any sense. 2%, 1% solids content, this is not a product. This is just you're spraying solvent, basically, dust. But that is actually a product. There are products that, phase 3 right now that are that dilute, but for that specific indication, for that, kind of indication that client is looking for, there is no other cure. There is no other drug out there that is available. So that makes sense to continue with that. But I agree that it is more of a perception, oftentimes at early stages.
Okay, let's give Firouz a round of applause for a wonderful talk.
Thank you.